morning, everyone, and thank you very much for joining us today at Breakfast at UM. My name is Nina Abdul, or Fadhlina Abdul Satar, a consultant clinical oncologist at UMMC. Today, I am here to introduce to you a name that needs no introduction, Professor Dr. Hogo Fuang. He is a well-known name in UM and in Malaysia. A clinical oncologist, was trained at Barts and the Marsden. He was the recipient of the Joint Commission International Outstanding Young Malaysian Award back in 2009 for medical innovation. His research interests involve breast, GI, and gynae cancers. He has also long championed participation in clinical trial and recently spoke at Cancer Research Malaysia Trial Connect 2022, both attended by Director General of Health and then Minister of Health. He is involved in many national and international collaborative research work. He is also council member of Together Against Cancer and champions cancer patients' rights in Malaysia. Today, he will be talking to us on clinical trials which are open in UMMC in GI malignancies. Prof. Ho, the floor is yours.
Thank you, Dr. Nina. So I have 30 minutes to talk about oncology clinical trials at UMMC. Actually, there are more trials than I can cover in half an hour. So we're only going to talk on GI and hepatobiliary malignancies. So thanks to my SC, Kimi, she has made this beautiful slide. So I think I'll give a quick overview of the studies that we run. So from molecular testing to stage 3 or below, first line, second line, and third line treatment, these are the tumor sites. So the tumor sites we're interested in actually include most of these sites, and we're interested in first line, second line head and neck cancer, triple negative breast cancer, adjuvant breast cancer at high risk for hormones. So either not positive or tumor more than 20% KICC7 or grade 3, patients who fail aromatase inhibitor adjuvant treatment for SERENA-4, and patients who have failed a couple of lines of chemo for oral. And lung cancer studies that we are particularly interested in stage 3 lung cancer, and also HER2 overexpressed lung cancer and PD-L1 more than 50% lung cancer, and also molecular testing for ALK fusion mutated lung cancer, and also general NGS for lung cancer. So we talk about GI malignancy in a minute. Prof. Manisa is interested in bladder cancer, stage 3 or below, and first-line renal cancer. And Dr. Rosita and Dr. Wan Samania are interested in gynae cancers and rare cancers. So we talk about gastric cancers. Now we have 2 recruiting studies. One is Fortitude and one is Horizon. So Fortitude is a randomized, multi-center, double-blind, placebo-controlled phase 3 study of bemarituzumab plus chemotherapy versus placebo plus chemo for patients with advanced gastric or gastroesophageal junction cancer with FGFR2b, fibroblast growth factor receptor 2b overexpression, Fortitude 101. So fibroblast growth factor receptor 2b is a member of the FGFR family, which contains FGFR1 to 4. It's a spliced isoform of FGFR2. FGFR2b is overexpressed is around 1/3 of HER2 negative gastric cancer. So the ligand, FGF7, 10, or 22, activate the FGFR2b receptor, causing dimerization, activate 2 possible downstream pathways, PIK-AKT, mTOR, or RAS NPK pathway, stimulating tumor differentiation and growth, tumor proliferation and growth. FGFR tyrosine kinase inhibitor has also been shown to be effective in other forms of gene derangement, like FGFR mutation, FGFR fusion, and translocation. So FGFR overexpression or amplification is associated with poor prognosis. So the different ways of gene derangement, which is protein overexpression, or activating mutation, or fusion, oncogenic fusion, causing basically overstimulation of the pathway, the MMPK and mTOR-AKT pathway. And the FGF can be excreted in a autocrine or paracrine fashion and stimulating angiogenesis and epidermal mesothelial formation. So this is what it looks like when IHC, so overexpress FGFR2b. This is what it looks like overexpressed on fish. So the study of Fortitude actually is based on the previous study, phase 2 study called FIGHT study, which is very similar design. So first-line gastric or GEJ adenocarcinoma with FGFR2b overexpressed either by IHC or by circulating DNA with the FGFR2 gene amplification. So for FOX versus for FOX plus bemarituzumab, 50 milligram per kilogram 2 weekly. So they reported a PFS benefit of 2.1 months, which is hazard ratio of 0.68, with reduction of disease progression and death of 32%, or overall survival benefit of hazard ratio of 0.58. So that is reduction in risk of death of 42%. So as we said earlier, activation of the FGFR2b pathway by the ligands stimulate the downstream mitogenic pathways. This can be inhibited with the FGFR inhibitor, bemarituzumab. So that blocks the growth factor signaling. But it can also causes the receptor degradation and causing antibody-directed cytotoxicity, killing by the immune cells, NK cells and macrophages by the ADCC mechanism. So design of Fortitude 101 is basically gastric or OGJ tumor with FGFR2b more than 10%, 2 plus or 3 plus, bemarituzumab plus 4-FOX or 4-FOX plus placebo. Second study is Horizon, which is zanidatamab plus chemo with or without a PD-1 inhibitor, tislelizumab by Novartis, which is a PD-1 inhibitor mainly developed in China for HER2-positive, unresectable, locally advanced or metastatic OGJ tumor. So background of HER2-positive gastric cancer or OGJ tumor came from TOGA study from 13 years ago now. So chemo plus trastuzumab versus chemo alone, giving a hazard ratio of 0.74, which is reduction of risk of death of 26%. So 13.8 versus 11.1 months. So that has been the gold standard for the past 13 years. A few efforts to improve the outcome. In fact, our center participated in Titan, Gatsby, and Jacob with addition of lapatinib or change it to TDM1 or addition of pertuzumab. But these studies have not been successful, although numerically there was some survival advantage, but did not reach statistical significance with the trial design. So those were not successful studies until the new concept of Keynote 811, which was published in the Nature December 2 years ago by Yelena Janjigian, comparing chemotherapy plus trastuzumab versus chemotherapy plus trastuzumab versus chemotherapy plus trastuzumab plus pembrolizumab, showing that the response rate goes up by 22% from 51.9 to 74%, and best response rate, which is complete response, also went up from 3 to 11%. So this is the early report from the Keynote 811, which is sort of changed the landscape, showing that HER2 positive gastric cancer also respond to PD-1 inhibitor. So the design of Horizon is similar along that line, which is chemotherapy plus anti-VEGF. Zanidatamab is a Beijing product, our first study with Beijing, a fast-growing China-US company, pharma. So chemotherapy plus zanidatamab and chemotherapy, zanidatamab is an anti-HER2 by Beijing. It's not very familiar drug to us. And this is the 811 design, which is chemo plus zanidatamab, anti-HER2, plus tislelizumab, anti-PD-1, which is the triplet, actually chemo plus 2 antibodies, and chemo plus trastuzumab as the standard of care. So that study is actually recruiting. So we're interested in gastric or OGJ tumor that overexpress HER2. Now to colon cancer. colon cancer, we have two studies recruiting. One is Crystal 10 and one is Origami. The second line is Origami is second, third, fourth line. Now it's going to come to second line soon. So it's many lines, but not first line. Crystal 10 is a study using adagrasib, MRTX849, with cetuximab, anti-EGFR antibody, versus chemotherapy for advanced colorectal cancer patients with KRAS G12C mutation who have progressed on first line of treatment. So the background about KRAS, RAS is the most frequently mutated oncogene in human cancer. The RAS oncogene are mutated in more than 20% of all cancers, including KRAS and HRAS. Fadhlina has the KURA study, which is HRAS inhibitor in head and neck cancer. KRAS is the most frequently mutated RAS family member. So more than 85% of RAS mutations are KRAS. And KRAS G12C, which is at the codon 12 location, glycine is replaced by cysteine, is found in 14% of lung cancer, lung adenocarcinoma, so it's more commonly seen in lung cancer, but also in 4% of colorectal cancer, but also in pancreas, gastric, uterine, gynae cancer, and cancer of unknown primary. So KRAS G12C is now druggable because KRAS traditionally has no pocket for them to design an inhibitor to latch on until they found that this switch 2 pocket in G12C that is druggable. So just a quick background about the RAS mutated tumor. So RAS GTP is the activated form of RAS, and GTP is the inactivated form, activated by upstream receptors signaling from, let's say, FGFR or EGFR. And when RAS is activated, it can activate the MAPK pathway, RAS-RAF, MEK, also the PIK3CA, MKT ATO pathway, and some rarer pathways causing proliferation, survival, and differentiation. So the G12C molecules looks like this, KRAS G12C. So that is the binding site of the ATP, of the GTP or GDP. And then the pocket of the cysteine sits here, switch 2 pocket. So you can design a molecule that can latch onto that position. Therefore, can inhibit it, stopping it from changing from active to inactive state, and so on. So it just lock it in an inactive state. So this is the kind of first generation KRAS G12C inhibitor. Now they're developing the second generation, which is located in a different activation state. So G12C inhibitor has been approved in the like sotorasib in G12C mutated lung cancer, but they found that it is not just in lung cancer that it works. It can also work in lung cancer is the green one, but also in colon cancer, can also work in pancreas cancer and cervix cancer. So that means if that is a driver mutation for that tumor, then it will work across a few tumor sites. For colon cancer, if you inhibit the RAS, actually there is one problem, which is the RAS-RAF MEK pathway. There is going to be a feedback or overexpression of the EGFR. So that means it will neutralize whatever you do. So that means if you want the KRAS inhibitor to work, apart from blocking the KRAS from activating, you have to inhibit the upstream pathway, that kind of vertical inhibition. So that means they think that it's better for it to work if you add in a EGFR antibody or a SHP2 targeted therapy. But SHP2 is still under development, so the more mature one is the anti-EGFR antibody like cetuximab or panitumumab. So it inhibit the upstream and inhibit the downstream KRAS, then it is more likely to work to block off the feedback mechanism of the inhibition. So Crystal 10 basically is a second line study. Patients had FOLFOX or FOLFIRI upfront, and you test them. You can pre-screen them when they're on their first line chemotherapy, and then if they have the G12C mutation, then they get recruited to have the second line chemo, which is for FOLFIRI or FOLFOX, depends on what's the first line. you can add in anti-VEGF as well, which is sponsored. So that means chemo plus bevacizumab as the control arm. The experimental arm is adagrasib, 600 milligram twice a day with cetuximab 2 weekly, 500 milligram per meter square standard dose 2 weekly. So this patient, this study, actually we have fully recruited our quota, but we have expanded the recruitment. So we are still looking for new patients with KRAS G12C mutation during first line to put them into the second line study. Origami 1 is a phase 1B, currently still phase 1B, not phase 2 yet, open label study of amivantamab, which is the it's a bispecific antibody against EGFR and MET versus nothing. So it is a single agent amivantamab for advanced colorectal patients who have failed irinotecan, oxaliplatin, and bevacizumab, or plus minus anti-EGFR antibody, i.e. it applies to people who have failed all the standard treatment, with the exception that you cannot have more than 3 lines of systemic treatment. So up to 3 lines of systemic treatment, they need to fail irinotecan, oxaliplatin, and bevacizumab at least. So just a background about amivantamab, also called Rybrevant. Just a piece of information, Rybrevant is probably now the most expensive oncology drug around, not counting the cell therapy. I forgot how much it is. It's just some ridiculous price because now it's a license for exon 20, EGFR exon 20 mutated lung cancer. So amivantamab is a bispecific antibody, which means that it can bind to 2 types of receptor, the EGFR receptor and also the MET receptor, and it demonstrates activity in inhibiting either receptors. So the preclinical data of the mechanism of action, which is the receptor binding and inhibiting the pathway, downstream pathway, receptor degradation, so it gets taken over by lysosome and degraded, and ADCC function, as we discussed just now with bemarituzumab. So the Chrysalis study is the amivantamab in exon 20 lung cancer. So they found good activity in lung cancer. So that led to the Mariposa and the Papillon study and led to the registration in lung cancer. So in the colorectal cancer model, they also found activity in different cell model. So of course, cell model is just to give you confidence that it will work because the cells wouldn't have an immune system and so on. So you still have to test it in human, which is what the phase 1B study is about. So this is what we are doing currently. So not the dose escalation bit. So we are doing the cohort ABC, which is of phase 2 cohort. So there are patients who had right side colorectal cancer or left side patients who have failed anti-EGFR, and then left side patients who have failed, as I say, oxali, irino, and bevacizumab. So they are going to open cohort D and E soon, and that would be in the second line setting, which is for FOXO4 plus amivantamab. So that one would be in the second line setting. So for these patients, they are all wild type patients, i.e. they are KRAS and RAS, BRAF wild type. Currently, they are talking about HER2 because there was a patient in the with HER2 mutation also did not respond to amivantamab. So in the future, they may also select out HER2 mutated patients. So this patient is still ongoing. So so we are still recruiting into this study. So for wild type patients, try to refer them because they get this one, which we have patients who have responded very well. So I think this drug is a good drug for wild type colorectal cancer. Now we have 10 minutes to finish off with the hepatocellular carcinoma. We have 3 studies for hepatocellular carcinoma, MRO3. This one is IMBRAVE 251 and DI0002. So MRO3. So for HCC, which we do MDT every Wednesday. So for early stage, you just ablate it, RFA, microwave, or resection. For intermediate stage, then you give TACE or you give SERT or some kind of embolization procedure. For first line, then it is the IMBRAVE 150 regime, which is atezolizumab, bevacizumab, but also Himalaya regime, which is durvalumab. This is a PD-L1 plus a CTLA-4 inhibitor, but you can still give the TKI, which is lenvatinib or sorafenib. In the second line setting, then you give either TKI, regorafenib, cabozantinib, or anti-VEGF receptor ramucirumab. You can still give nivo, ipi, or pembrolizumab, which is like 394 study in which we conducted a couple of years ago and found it to show survival benefit in a second line setting. So the Emerald study is for those with locally advanced tumor suitable for TACE. So the question is that whether TACE result of TACE can be improved by adding the Himalaya regime, which is the durvalumab plus tremelimumab plus minus lenvatinib. So lenvatinib is an interesting one because lenvatinib is the TKI, and lenvatinib plus pembro. Upfront, the study was not very successful, but this one we go one step further: lenvatinib plus CTLA-4 plus PD-L1 inhibitor. So yeah. So just go through the Himalaya study. Himalaya study not as famous as IMBRAVE 150, but actually it is it was reported last year, so it shows survival benefit also. The advantage of that is you don't have to give an anti-VEGF antibody, and actually it shows benefit by giving one dose of tremelimumab to kickstart the immune response, followed by durvalumab. So here is a survival benefit with hazard ratio of 0.78. So 3-year OS gone up by 10%, which is show the activity of the immunotherapy on some patients versus sorafenib. So tremelimumab, anti-CTLA-4 antibody, which is the one we talk about, that basically is one of the checkpoint. The first checkpoint that was drugged just before the PD-1 was inhibited. So Emerald 3 basically is for patients who are suitable for TACE. They are allowed to have one previous TACE, not suitable for resection or not suitable for RFA, microwave ablation, good liver functions, and no invasion of the portal vein, of the major portal vein. So Child-Pugh class A, good ECOG status. So they get the standard TACE, which is the standard of care, TACE plus the Himalaya regime, which is tremelimumab plus durvalumab. Durvalumab is monthly at 1,500 milligram, or the IO plus lenvatinib. Okay, lenvatinib 8 milligram or 12 milligram depends on their body weight. More than 60 milligram, 60 kg, they get lenvatinib 12 milligram. So that is what's the study? Emerald 3, which we have recruited very well. So we're going to increase the quota somewhat. So IMBRAVE 251 is a randomized study of atezolizumab plus lenvatinib or sorafenib in the second line setting, following failure of atezolizumab and bevacizumab. As we said just now, the practice-changing study, IMBRAVE 150, is basically giving atezolizumab plus bevacizumab. This is a PD-L1 inhibitor plus VEGF inhibitor, and found that and compared that to sorafenib, which is standard of care for about 15 years. And that showed significant survival benefit, hazard ratio of 0.66, risk reduction of death of 34%, survival 19 months versus 13 months. So that basically changed the practice overnight, and that becomes the standard of care, more adopted than lenvatinib, which show equivalence to sorafenib in the first line setting. So that means the standard of care, the preferred standard of care would be atezolizumab plus bevacizumab for the patients with advanced, not the locally advanced, but the advanced liver cancer. And TKI are indicated still, for example, if patients had very high risk of bleeding. But if they have very high risk of bleeding, TKI also increase risk of bleeding actually. So you may consider Himalaya regime for those patients. And then after the failure of atezolizumab bevacizumab, this second-line treatment that we talk about doesn't really apply anymore because they were done at the era of first-line was sorafenib. So that's where the data is lacking for this group of patients. Hence, the IMbrave 251 study was designed. So unresectable HCC progressed following previous atezolizumab versus lematinib. If they have atezolizumab, they can have lematinib, or lematinib plus atezolizumab, continue atezolizumab beyond progression. So that is the design of the study. I think this study we have fulfilled our quota, but we have expanded the cohort. So we are still recruiting some more. So that means if patients are on atezolizumab first line, we can kind of earmark them for recruitment to this study if they progress, or if they have stopped after 4 cycles, they progress, they can still be recruited. Last but not least is the BI-002 study of enzabendimab plus alone, or with the other one 880 and chemo, or cetuximab. For this one is for head and neck and for HCC. Okay, head and neck and HCC. Let's call it 002 study. So so the response rate for atezolizumab is around 30%, versus sorafenib about 11%. So complete response rate is not great, 5.5%. So the question is whether anti-VEGF plus checkpoint inhibitor is the way to go, because that's what the IMbrave 150 regime is about. So they think that it is we should make the tumor hotter by inhibiting CD47 sub SRP alpha interaction, because that may repolarize the tumor microenvironment towards a more pro-inflammatory phenotype, and causing more enhanced antigen presentation by lymphocytes, i.e. the innate response. I.e. it is not just about the adaptive response. The innate response trigger the adaptive response to be more effective by presenting the antigen and triggering the adaptive response. So it releases the brake on the phenotype of the lymphocytes, which are the cells for the adaptive response. So that means they use the anti-sub-alpha, which is BI-765063 enzalutamide, with the PD-1 inhibitor to try to create the additive or synergistic anti-tumor activity. So this kind of second generation immunotherapy. So we talk about the HCC first. So it's either they get the PD-1 inhibitor plus the sub-inhibitor, sub-alpha inhibitor plus PD-1 inhibitor, plus minus the anti-VEGF ANG2 inhibitor. So that is the first line for HCC. Second line is they get the PD-1 inhibitor, sub-alpha inhibitor, and the VEGF inhibitor, all at the same time. Okay, so that is the first line or second line HCC study. They also have a cohort for head and neck cancer in the second line setting using the 2 checkpoint inhibitors plus the antibody cetuximab. They don't use VEGF inhibitor for head and neck cancer for risk of bleeding. So versus plus chemo or plus cetuximab. So that will be the sub-alpha polymorphism patients who have B1 allele. That's the end of the talk. Thank you. I think we have 1 minute.
Thank you, Prof. for that talk. I think I invite questions from the floor, but my first question is we can see that there are a lot of molecules and drugs coming out from China, and they seem to dominate this area where there's fast drug development. How can we make Malaysia an attractive country for China to be doing their trials? Because we haven't really seen that many trials with Chinese compounds.
Definitely, yeah. So actually, China has many pharmaceutical companies. They just have not come to this part of the world. So I think to get more studies from China to here, first of all, we have to foster a good relationship. We have to perform on the Horizon study. If you do well and they think that you are a good site, your data is accurate and the clean up everything, then they'll give you more studies. So now Beijing has 2 studies in Malaysia. One is our Horizon. The other one is the first in human study in Kuching on the, that's Kuching is a US study. So that means they have 2 studies in Malaysia. I forgot what is the other one. So we just have to perform well on the studies, really. We have to recruit and make sure our data is accurate and clean.
And what do you think about our NPRA's views about registering Chinese compounds in Malaysia?
They have some difficulty with the, I forgot the the good manufacturing, GMP, and those kind of certificate. So that is the limiting factor. I think Malaysia NPRA has to open up a little bit, because currently they really just recognize the European and the American certificate. So, but they need to recognize that China is a big player and you cannot just like rely on the US and Europe forever.
Yeah, we have a question from the audience. I think it's from Kusara, Prof. Kusara. She says, glad to see that we are taking part in these international studies. So what about drug repurposing, really looking into older drugs in different ways, which will keep costs down, but won't make that much money for the pharma industry? Are we participating in that, and what's the landscape?
We are. We in ESMO this year in Madrid, we are going to see the result of adjuvant aspirin for colon cancer, the long-running study that I started when I started my career. So that was a 12-year study. So finally it ended this year. So we have some IIT. Usually they are IIT because of there's no pharma money in it. We are also looking at repurposing anti-helminthic drug with one of our collaborators. Forgot her name. What's her name? From St. George's and Royal Marsden Hospital in Yolanda. So that means we are doing, we're trying to do some drug repurposing. But as you know, to do studies, it costs money. And this kind of drugs are not that sexy. Pharma won't give you money, and FRGS think that you are too basic and not high-end enough, I think, to get the funding from FRGS.
Absolutely, we need to be pushing for this a lot more, because I think countries like ours will definitely benefit hugely from drug repurposing. So I feel that is all for today's session. Thank you very much for the questions, and thanks for presenting all the GI studies. So I invite all our colleagues to hopefully be aware of these studies. They're on Malaysian Oncology Society website as well. So if you are seeing a patient and you'd like to know which trials are open in centers in Malaysia, please visit MOS or Malaysian Oncology Society website to refer your patients. Thank you very much.
Thank you.